In this picture, there is an enzyme with an active open site. The substrate, in blue, then bonds with the active site and splits to form two new products. There is an enzyme with an open active site. In competitive inhibition, there is a competitive inhibitor in white along with a substrate in blue, both looking to bond with the active site. Then the competitive inhibitor binds to the site, which does not allow the substrate to bind. This shows that the substrate cannot combine because the competitive inhibitor is there. The substrate then moves on to find another enzyme. In non-competitive inhibition, there is an enzyme with an open active site and an open non-competitive inhibitor site. There is a substrate in blue and a white non-competitive inhibitor. The non-competitive inhibitor then bonds to its site as the substrate approaches. The binded non-competitive inhibitor closes the enzyme's active site, which does not allow the substrate to enter. The substrate leaves the closed enzyme, and this is non-competitive inhibition. In allosteric regulation, there is an enzyme with a partially closed active site and an allosteric site. There is also an allosteric activator in yellow and the substrate in blue. The allosteric activator binds to the allosteric site and opens up the active site for the substrate. The substrate binds to the active site and there is one product left. There is an enzyme with an open active site and an open inhibitor site. Substrate 1 in white binds to the active site. The product in yellow then becomes the substrate for enzyme 2. The substrate binds to the active site and the product leaves the enzyme. The product in green then becomes the inhibitor for enzyme number 1. Once the inhibitor binds to the site, the enzyme closes and the substrate cannot enter. This is feedback inhibition because the enzyme's final product becomes its own inhibitor.